Just keep in mind the composition beta. Okay, composition of both the layers, because this will help you to have the main differences between both of them. Okay. First is the presence and absence of an acid. And the second was what? Additional layer is present in which? Gram positive or gram negative?
Gram negative.
Gram
Yes, beta. We have an additional layer in gram negative ones. So the stain will easily penetrate in which one, according to you? Positive or
gram
negative?
positive.
Beta. Chali. Let's do this very quickly. We had done till here. Let's have it. Saffron in this. Conclusion now. Wall. In the positive, the wall is exposed, so it stains with the color. See, this stain, I'm talking about gram stain. Peptidoglycine is very adaptive to this stain. बहुत easily stain को ले लेता है बेटा. Which compound?
Peptidoglycine.
Peptidoglycine.
But the difference is when we talk about a positive one, the peptidoglycine wall तो बाहर ही है. So when you expose it or when you put, but in negative, what happens is the stain will not reach the peptidoglycine cell wall because of the presence of an extra layer. And extra layer does not contain peptidoglycine. What does it contain, beta?
Lipoproteins and polysaccharides.
Proteins and polysaccharides.
Very good. Safranin is taken by the outer layer, that is in the gram negative, because of the presence of lipoproteins. Like we said कि peptidoglycine बहुत जल्दी adapt कर लेता है to gram stain. Similarly, safranin को बहुत जल्दी adapt करता है, accept और absorb कर लेता है. What substance are we talking about?
Lipoproteins.
So lipoproteins very easily adapt safranin. So when you stain them with safranin, they will immediately give you red color. चलिए एक चीज guess करके बताइए before I show you. Which ones are more harmful for us? Gram positives or gram negatives? Guess.
Gram negative.
Why are they more you are absolutely right. Tell me, but why?
Since they have two layers, so they would be quite tough to be killed once they cause infection.
Very good, very good. Once they enter a body, a human body, for example, and some medicines are given, a treatment dose is given, then any particular medicine dose, chemical, drug, if we talk about a membranes to get affected. Yes, just one.
One.
And if it is two beta, so we'll have to think about a product which can pass through the outer membrane, which is made up of lipoproteins and
Polysaccharides.
very good. And after crossing that, it should also be able to pierce the inner membrane, which is made up of yes,
Peptidoglycine.
So when we talk about their harm to the human generation, then we will see this. You have already told me. Very good, children. You guessed it correct. Which ones are more harmful? More harmful, the gram negative ones, because drugs and medicines need to cross more barriers. For example, Vibrio cholerae. Vibrio cholerae is a gram negative bacteria. Anything you
Ma'am, it causes cholera.
very good. And when we say Vibrio, I'm sure you can guess the shape now, what or how it must be looking like.
Comma
You
shape.
remember Vibrio? Yes, this was the comma shape. Okay, let me very quickly I still have about 10 minutes. I'm talking about Moneras. Important features we already know. Let's classify bacteria on the basis of their survival in habitats. Name them very quickly.
Archaebacteria.
Adverse conditions are.
Archaebacteria.
So we have done two important members of Monera family, Archaebacteria and Eubacteria. Now name the third member of Monera.
Cyanobacteria.
Cyanobacteria or blue-green algae.
Yes, blue-green algae. How are they special? How are they different from other bacteria?
They are photosynthetic and they contain a green and a blue pigment.
Very good. They are able to photosynthesize. So now we are done with
Mycoplasma or
Mycoplasma or
PPLO.
PPLO.
what is the full form of PPLO?
Fluoropneumonia-like
Like
organisms.
organisms.
Okay, they are the smallest. Which kind of cells? Smallest cells with cell wall or without cell
Without
wall?
cell membrane.
Without cell membrane.
Mainly, how many members have you done, beta? In Monera
4.
till now? 4 you have
4.
done. Now let us classify them on the shapes. Name them very quickly.
Coccus, Bacillus,
Coccus,
Spirillum, Vibrium,
Bacillus,
and
Spirillum.
Mycelial.
Okay, beta. If we have two coccus together combined, what will you call them?
Diplococcus.
Diplococcus.
And if we have a chain of coccus, what is the other possibility that they can be found in a cluster also? So what are you going to call such cocci if you're going to find them in a cluster?
Staphylococcus.
Staphylococcus.
So we did the first classification on the basis of survival of habitat, Archaebacteria, Eubacteria. Then we did the second classification on the basis of the shape of the bacteria, where you had how many groups, Bache? How many types you had?
5.
The absence of flagella. If flagella is present, it's trichus. If it is absent, it is atrichus. Let me hide this. Can you name all the 5 types? Flagella present, the 5 types. You can do it together. Come on.
Monotrichus, Amphitrichus, Cephalotrichus, Lophotrichus, and Peritrichus.
4. We are left with 1. The last one was what, beta? Good. Then we did the structure of flagella. How many main parts of flagella did we have?
Basal
3.
body, hook, and filament.
Okay. So basal body is the one which is inside the membrane or it is outside?
Inside the
Inside.
membrane.
Very good. And the part which connects the filament to the basal body, what is it called?
Hook.
Very good. It is called hook. Filament is made up of which kind of proteins?
Flagellum.
Flagellum.
Globular proteins.
Yes, beta. It's a globular protein. And what is the name of the protein?
Flagellum.
Flagellum.
Good. We've done this also. Now let's go to this. We have two kinds of bacteria, gram positive and gram negative. The first structural difference between gram positive and gram negative, let's go to revise that. The wall is made up of, wall in both the structures is made up of
Peptidoglycan.
one acid. What is the name of the acid?
Teichoic acid.
Teichoic acid.
Good. And we have an additional membrane. In which one? Positive or negative?
Negative.
Negative.
That additional membrane is made up of what structure?
Polysaccharides and lipoproteins.
Yes, beta. So we are going to have an additional structure over here. So when we stain the process, you had this staining process, 90% gram stain. What are these bacteria called?
Positive. Gram positive.
And those which are
Gram
not equal to
positive.
stain beta, we can treat them with another stain. What is the name of that stain?
Safranin.
Safranin.
What color will you get with safranin with gram negatives?
Red.
Why are gram negatives easily adaptable or they can easily take this stain? Why? We had no beta. We are talking about the negative ones. Why gram negatives they easily become red? With
Because
safranin.
the lipoproteins present in their second or outer layer are quickly adapts or accepts the safranin.
Very good. And can you name the material which had easily adapted Gram stain?
Peptidoglycine.
Yes, peptidoglycine. But in gram negative, peptidoglycine could be taken. Which ones are more harmful for health?
Gram negative.
Gram negative.
And what is the reason?
Because they cannot be killed
Because they
easily.
are bilayered, so drugs and medicines do not work on them.
It's difficult. It takes more time, and we need more advanced kind of drugs to treat such bacteria. We just did an example of a harmful gram negative bacteria.
Vibrio cholerae.
Diseases or disorders which are caused by gram positive or negative?
Negative.
Gram negative.
Gram negative bacteria. Till here, everybody is okay with Moneras?
Yes, ma'am.
Sure.
Yes, ma'am.
One last question before you leave, beta. 4 main members of Monera.
Archaebacteria,
Archaebacteria,
Eubacteria,
Eubacteria, Cyanobacteria, and
Mycoplasma.
Mycoplasma.
Good. We'll continue tomorrow. Tomorrow